What happens to the fat you eat? How does it travel through your body, and why is cholesterol so important? Understanding lipoprotein metabolism is essential for medical lab scientists, biochemistry students, and healthcare professionals. In this video, we'll break down 4 critical pathways: how dietary fats are absorbed, how the liver processes lipids, how LDL, often called bad cholesterol, is removed, and how HDL helps keep your arteries clean. When these pathways malfunction, the risk of heart disease and metabolic disorders rises. Stick around as we simplify this complex topic to help you master lipid metabolism. The first pathway in lipoprotein metabolism is the intestinal, or exogenous, pathway. This pathway is responsible for absorbing dietary lipids, primarily triglycerides and cholesterol, and delivering them to the bloodstream. It all starts when you consume fats. In the small intestine, dietary triglycerides and cholesterol are emulsified by bile acids, making them easier to digest. Then, pancreatic lipase breaks down triglycerides into free fatty acids and monoglycerides, which are absorbed by intestinal cells, known as enterocytes. Inside these enterocytes, triglycerides are reassembled and packaged into large lipoproteins called chylomicrons. These chylomicrons contain 90% triglycerides and small amounts of cholesterol, along with important proteins like ApoB48. Before entering circulation, chylomicrons pass through the lymphatic system and then enter the bloodstream via the thoracic duct. Once in circulation, chylomicrons interact with high-density lipoproteins, HDL, acquiring 2 key proteins, ApoC2 and ApoE. ApoC2 is essential because it activates an enzyme called lipoprotein lipase, LPL, which is attached to the inner walls of blood vessels in muscles and fat tissue. When LPL is activated, it hydrolyzes triglycerides inside chylomicrons, releasing free fatty acids. These fatty acids are either used for energy in muscles or stored as fat in adipose tissue. As triglycerides are removed, chylomicrons become smaller, turning into chylomicron remnants. These remnants are rich in cholesterol and are taken up by the liver, where they bind to receptors via ApoB48 and ApoE. Once inside the liver, chylomicron remnants are broken down, and their cholesterol is used for bile acid synthesis, new lipoprotein formation, storage or secretion into bile. Interestingly, about 85 to 90% of cholesterol in the intestines isn't from food but comes from bile and other internal sources. The absorbed cholesterol can be transported back into chylomicrons or converted into nascent HDL through an important protein transporter called ABCA1. Finally, dietary triglycerides that remain in circulation undergo a lipid exchange process, where they are transferred to other lipoproteins like VLDL, IDL, LDL, and HDL. This process is mediated by cholesterol ester transfer protein, CETP, and helps distribute dietary lipids throughout the body. In summary, the intestinal pathway is crucial for transporting dietary fats. It starts with digestion and absorption, continues with chylomicron formation and circulation, and ends with the liver breaking down chylomicron remnants. This pathway ensures that dietary lipids reach cells for energy, storage, or further processing. But what happens next? Now that dietary fats have reached the liver, how does the body manage internally produced lipids? In the next section, we'll explore the hepatic, or endogenous, pathway, which controls how the liver processes and distributes fats. Unlike the intestinal pathway, which deals with dietary fats, the endogenous pathway manages lipids produced by the liver and distributes them throughout the body. This is crucial for maintaining energy balance, cholesterol homeostasis, and cell membrane integrity. The liver is the central hub for lipid metabolism. It synthesizes, stores, and exports fats as needed. One of the key players in this pathway is very low-density lipoprotein, VLDL, which carries endogenously synthesized triglycerides and cholesterol from the liver to peripheral tissues. VLDL formation and secretion. In the liver, triglycerides and cholesterol are packaged into VLDL particles. This process requires microsomal triglyceride transfer protein, MTP, which helps incorporate lipids into ApoB100, the structural protein of VLDL. Without MTP, VLDL cannot be properly assembled, leading to conditions like abetalipoproteinemia, where ApoB-containing lipoproteins are absent. Mature VLDL particles contain 55% triglycerides, ApoB100 as a structural component, ApoC2 and ApoE, acquired from HDL after secretion. Once secreted into the bloodstream, VLDL particles circulate and encounter lipoprotein lipase, LPL, which hydrolyzes their triglycerides. This process releases free fatty acids, which are then used as an energy source in muscles or stored in adipose tissue for future use. As triglycerides are removed, VLDL shrinks and becomes denser, transforming into intermediate density lipoprotein, IDL. VLDL to IDL and LDL conversion. Not all IDL remains in circulation. About 50% of IDL particles are taken up by the liver through hepatic remnant receptors, which recognize ApoE. The remaining IDL undergoes further lipolysis, where cholesterol ester transfer protein, CETP, facilitates the exchange of triglycerides for cholesterol esters, converting IDL into low-density lipoprotein, LDL. LDL is cholesterol-rich and serves as the primary carrier of cholesterol to peripheral tissues. Cells with LDL receptors recognize and internalize LDL particles, allowing them to incorporate cholesterol into cell membranes, use cholesterol for steroid hormone synthesis, store excess cholesterol as cholesterol esters. However, excessive LDL levels can be problematic. If not efficiently removed, LDL particles can infiltrate blood vessel walls, leading to atherosclerosis and cardiovascular disease. This is why LDL is often referred to as bad cholesterol. It plays a key role in the formation of plaque buildup in arteries, increasing the risk of heart disease and stroke. Cholesterol management in the liver. The liver plays a critical role in regulating cholesterol levels. It obtains cholesterol from three main sources. 1. Dietary cholesterol, delivered by chylomicron remnants. 2. Synthesized cholesterol, via the HMG-CoA reductase pathway. 3. Uptake of LDL particles from circulation. Once inside the liver, cholesterol is utilized in several ways. It is incorporated into VLDL and secreted back into circulation. It is converted into bile acids, which aid in digestion. It is stored as cholesterol esters for future use. The liver also regulates cholesterol production through negative feedback. When intracellular cholesterol levels are high, the liver suppresses HMG-CoA reductase activity, reducing cholesterol synthesis. Reduces LDL receptor expression, limiting cholesterol uptake. Increases bile acid synthesis to enhance cholesterol excretion. This delicate balance ensures that cholesterol homeostasis is maintained. However, when this regulation fails, such as in genetic disorders like familial hypercholesterolemia, cholesterol levels can rise uncontrollably, leading to a high risk of heart disease. Comparing VLDL and chylomicrons. VLDL and chylomicrons share similarities but differ in key ways. VLDL originates from the liver, while chylomicrons come from dietary fats. VLDL triglycerides are hydrolyzed more slowly than chylomicrons. Chylomicrons have ApoB48, while VLDL has ApoB100. VLDL has a longer circulation time compared to chylomicrons. In summary, the hepatic pathway is essential for transporting liver-produced lipids. VLDL delivers triglycerides to tissues, transforms into LDL, and helps regulate cholesterol levels. The liver ensures that lipid transport is balanced by controlling cholesterol synthesis, storage, and excretion. But what happens when LDL levels rise? In the next section, we'll explore the LDL receptor pathway, the body's main mechanism for clearing LDL from circulation. This pathway is essential for cholesterol homeostasis, as it controls how much LDL remains in the bloodstream. LDL uptake by cells. LDL particles circulate in the blood and eventually bind to LDL receptors on cell membranes. These receptors are found in many tissues but are most abundant in the liver, adrenal glands, and peripheral cells that require cholesterol. The LDL receptor specifically recognizes ApoB100, the primary protein on LDL. Once bound, the LDL particle is taken into the cell via endocytosis, forming an endosome. Inside the cell, the LDL is transported to the lysosome, where it is broken down. Fate of LDL components. Once inside the lysosome, ApoB100 is degraded into amino acids. Cholesterol esters are hydrolyzed into free cholesterol. This newly released cholesterol has several important functions. It is incorporated into cell membranes. It is used for steroid hormone synthesis, in adrenal and gonadal tissues. It is converted into bile acids, in the liver. However, excess free cholesterol can be harmful. To prevent overaccumulation, cells activate negative feedback mechanisms that 1. Reduce LDL receptor synthesis, decreasing further LDL uptake. 2. Inhibit HMG-CoA reductase, lowering cholesterol production. 3. Activate ACAT, acyl-CoA cholesterol acyltransferase, which converts free cholesterol into cholesterol esters for storage. Role of PCSK9 in LDL receptor regulation. A protein called PCSK9 plays a crucial role in LDL receptor recycling. With PCSK9, LDL receptors are targeted for degradation, reducing the number of available receptors and increasing LDL levels in the blood. Without PCSK9, LDL receptors are recycled and reused, allowing for more LDL uptake and lower cholesterol levels. This is why PCSK9 inhibitors are an effective treatment for high cholesterol. They prevent LDL receptor destruction, enhancing cholesterol clearance from the bloodstream. LDL and atherosclerosis. When LDL is not effectively cleared, excess LDL can accumulate in blood vessel walls, leading to atherosclerosis. LDL can undergo oxidation, triggering an immune response where macrophages engulf oxidized LDL, forming foam cells, a key feature of arterial plaque buildup. This process increases the risk of heart attacks and strokes. In summary, the LDL receptor pathway is critical for maintaining cholesterol balance. LDL receptors regulate how much LDL is removed from circulation, while PCSK9 controls receptor recycling. When LDL levels are too high, atherosclerosis can develop, increasing cardiovascular disease risk. But how does the body counteract excess cholesterol? In the next section, we'll explore the HDL-mediated reverse cholesterol transport pathway, which helps remove excess cholesterol from the bloodstream. This process is critical for maintaining cholesterol balance and preventing atherosclerosis. Formation of HDL. HDL begins as a small, immature lipoprotein called nascent HDL, which is mainly composed of apolipoprotein AI, ApoAI. This protein is secreted by the liver and intestines and serves as the foundation for HDL maturation. In circulation, HDL interacts with cells via the ABCA1 transporter, which allows it to pick up excess free cholesterol and phospholipids from peripheral tissues. As cholesterol accumulates, HDL particles become larger and more spherical, transitioning from nascent HDL to mature HDL. Cholesterol esterification and transport. Once inside HDL, free cholesterol is converted into cholesterol esters by the enzyme lecithin cholesterol acyltransferase, LCAT. This modification allows cholesterol to be efficiently stored within HDL's core. Mature HDL then delivers this cholesterol ester to the liver and steroidogenic tissues through two primary mechanisms. 1. Direct transport via scavenger receptor B1, SR-B1, HDL binds to SR-B1 receptors in the liver, where cholesterol is selectively extracted for bile acid synthesis or excretion. 2. Indirect transport via cholesterol ester transfer protein, CETP, HDL transfers cholesterol esters to LDL and VLDL in exchange for triglycerides, allowing LDL to deliver cholesterol back to the liver. HDL's antiatherogenic role. By removing excess cholesterol from arterial walls and foam cells, HDL helps prevent plaque formation, reducing the risk of cardiovascular disease. This is why HDL is often called good cholesterol. In summary, the HDL-mediated reverse cholesterol transport pathway helps clear cholesterol from peripheral tissues and delivers it to the liver for excretion. This process plays a crucial role in cardiovascular health and cholesterol homeostasis. Lipoprotein metabolism is a complex but essential process that keeps our cells functioning and our cardiovascular system healthy. We've covered the four major pathways: intestinal, exogenous, pathway, which absorbs dietary fats; hepatic, endogenous, pathway, which transports liver-derived lipids; LDL receptor pathway, which regulates cholesterol uptake; and HDL-mediated reverse cholesterol transport, which removes excess cholesterol. Understanding these pathways is crucial for medical professionals, as imbalances can lead to heart disease, metabolic disorders, and other health issues. If you found this video helpful, be sure to like, subscribe, and share for more in-depth explanations on medical and biochemical topics.